Hello.
Hello. We are waiting for the prorector and then we'll start. Yes.
Okay.
Please go ahead, prorector.
Okay, thank you. Hierbij I open this academic ceremony in which Congying Zheng will defend the academic thesis optimizing the comet assay to measure DNA damage and repair. May I invite you to present a summary of your thesis and the conclusions of your thesis.
Okay. Thank you, Pro-Rector. I will share my presentation to all esteemed members of the Corona. Can you see my sharing?
Yes. Go
Okay.
ahead.
Good morning, oh, sorry. Wait a second. Stop share. Good afternoon, dear Prorector, esteemed members of the Corona, supervisors, colleagues, beloved friends, and my dear friends. Thank you all being here to share this significant milestone with me. Over the next 15 minutes, I will introduce you a fascinating method known as the comet assay, and it's optimized to measuring DNA damage and repair. Our human beings are exposed to a lot of the bad hazards, such as air, water, contamination from industry, or improper intake of the drug and treatment, or unsafe food. Our DNA will be targeted and attacked by these exposures. Most of the time, our body can keep balance between the DNA damage and the DNA repaired. So it's fine. We are happy and healthy. Unfortunately, if there is unbalance between damage and repair, cancer disease will eventually occur. Here, I guess you are confusing about what the roles of the DNA in our human body. Just the image that our body is a house constructed from a lot of the bricks. DNA represent the individual bricks. One day, some bricks, the DNA, are damaged and far out of the place. The DNA repair act like a bricklayer. He would replace the damaged brick, restoring the house to its original condition and functionality. However, if this bricklayer is unskilled or lazy, the house will eventually fall into disrepair and become a ruin in the long term. Thus, it's necessary to have some fast, sensitive, high-throughput methods which could measure the DNA damage and repair in the beginning when we exposure to this contamination hazards. In these assays, cells has been loaded in the gel after electrophoresis. The damaged DNA will be migrated from the DNA supercoiling and make a tail. The shape is similar as a comet. That's we call it comet assay. You can spot the vertical part, the middle vertical part. A longer tails means higher DNA damage. That means the cell's exposure to very high DNA damage. In the right images, for the DNA repair assays, the comet has a longer tails at the beginning and monitoring over time the time courses. The tail decrease over the time courses. In a word, faster tail decrease than a higher DNA repair. With the advancement in the comet scientific community, most studies so far utilize the 12 gel or 90 gels version, moving away from the traditional 2 gels version. Well, this has significantly increased the efficiency, but we don't stop here. We are developing an automatic system using robotic and the AI system. The system will not only run the assays, but also incorporate a cloud-based system for scoring and provide the expert suggestion for the human biomonitoring. The more interesting things and surprise me, the new system can process the 482 samples per hour, totally hands-off. So far, this comet assay and its modified versions has been widely applied in various fields. Most notably, it's used in the human biomonitoring to assess the environmental and occupational hazards. Moreover, in epidemiology studies, it helps us to understand the how host-related factor influence on the DNA damage or repair, and to determine whether nutrition interventions is effective or not. The assay is also utilized in lab experiment to investigate the dose-response relationship and make the risk assessment for unknown chemicals. In the end, but not least, it has clinical applications such as cancer trials could be a predicted biomarker for the tumorigenesis, and a diagnosis to assess the male fertility and infertility in human reproduction. And for my thesis, many focus on 3 areas, which are highlighted by the red rectangle: human biomonitoring, epidemiology study, and nutrition intervention. My thesis has been divided into 2 parts. The first part is how to improve methods for DNA damage assessment. In the paper 1, a rain trial study has been collaborated by 13 investigators from 11 labs. The central lab at Copenhagen University delivered the comet image to collaborating partner for measuring the DNA damage by either using the visual scoring or semi-automatic scoring in the comet assay. We aim to revise the visual scoring in the comet assay and provide guidelines for the visual scoring relative to the image-based scoring. Well, we found inter or intra-scoring variability in visual scoring of the comet assay and concluded that future standardization of the visual scoring is desirable. Nevertheless, the 5 classic visual scoring methods remain a reliable procedure for measuring the DNA migration. In the second one, another rain trial study was conducted by 8 labs from H-comet network. Cells treated with different concentrations of the potassium bromate has been frozen in -18 in the central lab Copenhagen University and delivered to different labs. Virals from the same batch of the cells were analyzed once per week for 3 consecutive weeks, round 1. And keep the cell frozen in 2 years, 3 years. They are just round 2 and round 3. And did the comet assay in the year 2 and year 3. The samples in the round 1, X1 to X3, is both inter-assay variation and the putative systematic effect of the cryopreservation. The results show that potassium bromate is a suitable positive form-emitting DNA glycosylase, we call it FPG, modified comet assay positive control, and provide our reliable results. The second part is how to improve the methods for measuring DNA repair. In the paper 3, we adapt a cellular repair assays based on the FPG modified comet assay to assess the DNA repair kinetics. The removals of the oxidatively damaged DNA over time, 1 hour, was analyzed in many cell types. The RO compound +9 induced the initial amount of the damage virals between cells, and this was apparently associated with the rate of the repair of these cells and the antioxidant ability. We found that potassium bromate could induce similar initial amount of damage at the beginning, which allows to accurate analysis of the subsequent cellular repair kinetics in first hours. You can see in the first beginning, 0 minutes. There is a comet with a longer tails and the following with the time. That means faster decrease of the comet tail, higher DNA repaired, vice versa. In human biomonitoring study, most samples are frozen, which makes them difficult to process and laborious to handle. to simplify the frozen sample processing and improving in efficiency. Modified versions of the comet assay called in vitro repair assays have been developed. It has been described in the figure 2. Meanwhile, both assays, the figure 1 and the figure 2, have been used and applied to measuring the DNA repair for many years in human biomonitoring. There is no direct comparison between them. Unlike the cellular repair assay, where living cells are exposed to DNA damage compounds and monitored over time, in the in vitro repair assay, the cell extract will take from the tested cells and keep in the -80. You can do whatever whenever you want and treat it with the substrate nucleus with the targeted DNA. And the longer tails means longer incision activity. But this finding suggests that in the end, these two methods should be viewed as a complementary tools rather than interchangeable methods, because cellular repair assays measuring the whole process of the DNA repaired and in vitro repair assays only measuring the incision process of DNA repaired. In paper 4, a broad database of the study from H-comet network was assembled and analyzed on the nucleation repair activity. Just to think about the nucleation repaired as your cells as a repair security guard. When it works, your body might handle environmental toxin or agent better. Nucleation activity data in PBMC will normalized and correlated with virus host factor, gender, age, BMI, and smoking habit. In this multifaceted analysis, undiscovered very significant sex disparity. It means significant higher nucleation repaired in female compared to the male. Higher nucleation repaired in older subjects, especially for the oldest lady, age older than 17 years old. Most precisely, female with normal BMI shows the highest levels of the nucleation repair, to be specific, whereas the lowest nucleation repair was observed in the male with smoking habit and with obesity. This type of people should be taken care. In the last paper, we employed the in vitro repair assay to measure the basic exhaustion repair in PBMC from the colorectal cancer patient, follow a 6-month food intervention. The control group only received a standard care advice. And for the intervention group, they received anti-inflammatory and antioxidant-rich food for 6 months. But unfortunately, the results do not support the effect of the dietary intervention on basic exhaustion activity in post-surgery for colorectal cancer patient. Well, diet didn't affect the basic exhaustion repair in the study, but it helped us to understand what doesn't work, so we can focus on finding better ways to support recovery. So by now, I think you should have a clear understanding of how the comet assay and its modified version to applications across the virus field. We considering the optimized comet assay with visual scoring to be a sensitive and reliable method for measuring DNA damage, particularly in human biomonitoring study. Furthermore, the DNA repair measured by enzyme-modified comet assay as a promising biomarker in health research, providing a sensitive and reliable results. In the end, a stunning images I put here capture a night sky with a vibrant aurora and a bright comet, which glows brightly with its tail striking across the sky, adding a celestial charm to the scenes. I must say in the end, the comet assay truly is a heavily method in the health research. Thank you.
Thank you for your presentation. The opposition will be opened by Dr. Boots. Dr. Boots is assistant professor in redox toxicology at the Maastricht University, and she was member of the assessment committee.
Okay. Thanks, Pre-Rector.
Dear candidate, As a start, I first would like to congratulate you on the completion of your dissertation. And of course, I would like to extend my congratulations to your whole team, your whole supervisory team. I've read the whole manuscript as part of your assessment committee with great interest and growing enthusiasm. And I'm impressed by both the quality as well as the quantity of the work that you have done. As you also shown now in your presentation, you have 6 scientific papers, chapters, of which 5 have already been published. And I think that's a great achievement, especially if you consider, as you say it in your own words in the acknowledgements, that you started with awful English at the beginning. I think it's a great accomplishment, what you have shown. As you might know, my research interest lies within redox biology, so I think it will not be a surprise. I would like to go to chapter 4. To discuss with you what you have investigated there in the repair of oxidized nucleobases. And you have applied the FPG modified comet assay in that chapter to assess the DNA repair kinetics. And since you did not really explain it, I would just like to start off easily to ask you for a short reminder on exactly the working mechanism of the FPG modification in the comet assay. So could you briefly explain the working mechanism? So what is FPG doing specifically in your comet assay?
Okay, dear esteemed opponent, thanks for your question. And for the FPG modified comet assay, the FPG is just the enzyme. If the DNA repaired glycosylates could be recognized 8-oxyguanine and also the FPG and reopen the purine. So it can just recognize this type of the lesion, oxidative lesion.
Okay, yeah, so it cleaves the purines on the oxidative damage. Yeah.
But many, 80% are 8-oxyguanine in this one.
Yes, I agree. And then you have used 2 different components to induce the oxidative damage. So you've used Rho and light, and then also potassium bromate. I was not familiar with potassium bromate, so I looked it up. I found out it's a carcinogen in Europe, yet it's still used in the US. So please be careful now, because I understand you're living there now. They still use it in bakery products. And you said that it's not very clear what the working mechanism is. So on page 1112, you don't have to look it up. You say it's not really understood how it works. And then luckily at page 123, you state that glutathione is involved. And then you got me curious, but I couldn't find the reference. So I was wondering, do you know, or could you tell me, how is potassium bromate inducing oxidative damage to your DNA? What is happening with this trigger? Do you know that?
Yes, thank you for the question. And these mechanisms of the potassium bromate to induce the X-oxyguanine is dependent on the one electrosion dependent methods. It means when you treated the cells with potassium bromate, it can make the bromine radical, and this can just abstract an electrons from the guanine out. And this residue will react with water to make the 8-oxyguanine. That's it.
Okay. No, wow. I didn't need a reference. You know it by heart, so that's great. And then could you tell me, what is then the role of glutathione in this process?
Sorry.
You state in your thesis that glutathione, so GSH, is needed for the effect of potassium bromate. What is the role of the GSH in this damaging process?
Actually, the GSH is a type of the cyan. It's a type of the cyan. So, but it's interesting for the potassium bromate. It's doing the depletion of this GSH antioxidant. So that's the reason for when we apply the potassium bromate, this potassium bromate treated cells was not influenced by the antioxidant. That's the reason. But for for the root powder, right, +9, this one, the initial amount of the damage has influenced by the rate of the repair of the cells and also antioxidant ability of the cells. We did this experiment. We find the difference for these 2 chemicals.
Okay. Well, I'm very happy that you go into that part, because that would be my next question. So you gave me a perfect bridge to my next question, because I was looking into GSH, and then you say on one hand, GSH is helping potassium bromate to induce more damage. And on the other hand, GSH, of course, is a very potent scavenger. It's an antioxidant, and we need it in our body. And then I saw in your methods that you are doing an antioxidant status analysis. I was, as a redox toxicologist, I was happy to see that. But then the assay is not actually measuring antioxidants. What you're doing in the assay is checking how your cells react on hydrogen peroxide. Why did you choose this assay? Why did you not just measure your full antioxidant level?
It's very important because there already have 2 papers published around 2006 by my supervisors, Andrew Collin. They are just treating with hydrogen peroxide. So it reflects the antioxidant. It is a challenging assay.
Yes, it's a challenge. I agree with that.
Yeah.
Hydrogen peroxide for sure is a challenge on your cells.
Yeah.
It's not necessarily, I think, reflecting your antioxidant status. You could have also maybe measured your total antioxidant capacity, or you could have measured maybe glutathione levels.
Definitely agree with you. Probably to be more specific, we can just measure the levels of the GSH for cells. But for us, it's for the beginning. We are just trying to apply the methods for the human biomonitoring. So we are just trying to do some simple experiment, to make it easier for, for the technicians in the public health and epidemiology part. Probably the lack of the equipment or some device to do it. So we just believe about you treated with cells with hydrogen peroxide can reflect the total abilities of the antioxidant. That's it.
Oh, I agree. I agree on that part. I would not call it antioxidant status, but I definitely agree with you that challenging your cells with hydrogen peroxide will give you a clue on how well the antioxidants are functioning. Yeah, I agree with that. And then I'm very shortly going to I'm going to look at the time. Yes, I'm very quickly going to ask. You compared both of the triggers, so you did Rho and you did potassium bromate, and then you first looked on cell viability. You did that on page 131, and I was surprised to see. So if you treat your cells with Rho, your cell viability stays more or less similar. You go from 95% to 92%.
Yeah.
If you treat with potassium bromate, you go from 95 to 82%. I couldn't see whether you checked here for statistical significance. I would argue from 95 to 82% is quite a decrease in cell viability. And of course, if you would have apoptosis, that would influence your measurements. So I was wondering, did you check, is this cell viability significant different upon treatment?
No, because they have a very high standard deviation. We tried to do the statistic analysis between this 95 and 80% viability, but they are still without statistically significant. And by the way, I just could be clarified because when I doing this experiment, it's not I doing one cells and one by one. It's I just run all the cells in parallel. To avoid some bias or something. So could be the cells is just in the last one. So could be some simple process problem here.
Okay, that makes sense. Okay, then I'm very satisfied with your answers, and I would like to hand proceedings back to the prorector. Thank you.
Okay, thank you.
The opposition will be continued by Professor Wilson, professor in neuroscience at the Hasselt University. And Professor Wilson was also member of the assessment committee.
Yes, thank you, Mr. Candidate, for the rather enjoyable and insightful presentation. I very much enjoyed it. I thought it was a really nice encapsulation of the work that you've done and the advances that you've made. I have a few questions here. Let me quickly drag up this file. So you've described, you sort of focused most of your thesis around optimization of the COMET assay, and I think that's where most of your advances have been made. And you compare in one chapter the cellular repair assay, or this challenge assay, with the in vitro repair assay that you've also described was extract-based. So as an expert, and I consider you now an expert in running these assays, if I was to come to you and ask you which assay I should employ to do a population studies, what are some of the key elements that you would suggest that I consider in determining whether or not I use a cellular repair assay and in vitro assay? What are the strengths of them? What are the weakness of them? And how can they be differentially applied? I understand they're complementary, but they're really assessing two very different things. So I'd be interested in hearing what your evaluation is of the two assays and how they can be applied in human studies.
Okay, dear highly esteemed opponent, thanks for your question. Well, actually, I like your questions, and I read a lot of your comments in my thesis, actually. And for this one, I had to see why you choose. If you just these guys come to me and ask me which one is better, I had to see it's dependent on your study purpose and the purpose. If you just go for the human biomonitoring, I suggest to go for the in vitro repair assays because it can just give you some information about the exposure incision activity. And by the way, the in vitro repair assays is applied for the frozen samples. It's very good for the human biomonitoring, especially when they're handling 100 or 200 samples per day. So in vitro repair assays will be amazing things. But if you just go for the clinic treatment or just want to know the whole cellular kinetics after treated with some drug or treatment nutrition study, I would suggest to go for the cellular repair cellular repair assays.
Yeah, that's nice. I like that idea. So building on the in vitro repair assay, indeed, what you're assessing is essentially a single step in the process. That's the initial cleavage event by the glycosylase, or perhaps a combination of the glycosylase and endonuclease. I'm wondering if you've considered expanding your in vitro repair assay to incorporate the other steps of the process. I mean, there's a gap filling step, as you well know, and then there's a Nick ligation step. Can you envision an experimental assay, which is an in vitro repair assay that captures the entire base excision repair response as opposed to just a single initial step? And what might that assay look like?
Well, actually, this one, you know what, I already talked with some very expert, and we are going to try this experiment in my postdoc. But I also see this comment from you in the thesis, but probably we have a different logic because you are going to start from the APE and the polymerasis one from the beginning, right? But from my side, I prefer to treat it with the in vitro repair assays over the time courses, longer time courses, to see whether the comet tail decrease or not. If the comet tail still keep the same, that means the lack of the steps of the basic exertion repair, right? So I will start from the end step. I will aiding the ligation with ATP to see whether they have the comet tail decrease or not. Just to go step by step. And for the APE and the APE step, I will try to using the difference characteristic of the monofunctional enzymes or biofunctional enzyme, because for the monofunctional enzyme, they don't need APE1, right? So I will try to see the difference. Moreover, using some MEF knockout gene is fantastic for this experiment. We can just try the MEF APE1 knockout cells or polymerasis 2 knockout cells to see which step really working in the incision repair activity. That's it.
Yeah, indeed. And some of that work is ongoing, as you probably are aware. So my last question is related to this idea of dietary interventions, and I think that's been a major focus, and it's also related to BMI potentially influencing the outcomes related to repair capacity. So your data showed really that the dietary intervention, or in this case, the antioxidant supplements, didn't seem to have a consequence on DNA repair capacity. But is there evidence that dietary interventions or some anti-aging compounds or something that currently is present within society actually has a stimulatory effect on DNA repair capacity? Are you aware of that? And because I'd really like to know your insights in terms of what is known about interventions and stimulation, or potentially even, I guess, abrogation of DNA repair capacity.
definitely some dietary interventions could modulate the basic exertion repair. For example, like Andrew Collins did the kiwi study for the using the crossover trials, to try to the kiwi food can just modulate the basic exertion repair activity. Moreover, they also have some broccoli intervention. They can also just modulate the nucleation repair, but it's controverted because it's based on the type of the food.
And so what would you recommend if you were to operate the Food and Drug Administration in some country? What would be your recommendation for what supplements or what food should I eat in order to enhance my repair activities?
Well, just to try to get more antioxidant rich food and anti-inflammation food, or a kiwi, definitely. Every day we are just make a joke to try to get more kiwi.
Yeah, I'm gonna go to the store and get kiwi now. But
yeah, thank you.
All right, thank you very much. That ends my questions, Mr. Candidate.
Okay.
Thank you. The opposition will be continued by Professor Dag Anders Brede, professor at the Norwegian University of Life Sciences. Professor Dag Anders Brede was also member of the assessment committee.
Okay, thank you. So I would like to start by congratulating the candidate on a very well written thesis and a good piece of work. So I'm an ecotoxicologist working on effects of ionizing radiation and heavy metals. And I've used the COMET assay for a number of different organisms, from nematodes to zebrafish and other types of fish and mice and plants even. And in my experience, COMET assay is really delicate. It's sensitive to a lot of different steps and can have a lot of variation. Could you summarize the most important things to do in the lab to avoid variation? Because you probably have the best experience in optimizing this assay of all of us.
Okay. Dear highly esteemed opponents, thanks for your question. Well, I had to see before you're doing the COMET assay, it's better to read two natural product paper. One paper published by Andrew, 2023, right? Another one is called mini information of the COMET assay report. They will just show us how to minimize the variations of the COMET assay. But from my experiences, I had to see from the beginning, the sample process, especially from the frozen, is very important. And then just go for the COMET assay process is in the electrophoresis. The temperature control and the duration is very important. After that is the staining process. The staining is also good influence factors for the COMET assay, as we did for like the chapter 2 and the chapter 3, right? We did this rental study. We found this staining process could be the main reasons to make the variations for the COMET assay. Thank you.
So I was very impressed by the result of these ring tests that you have done. I mean, all of these labs have really high quality in their results. That's obvious. But when you see other papers around, I mean, there is a lot of variation in the results from different labs. And do you consider COMET a really robust assay? Or when you are reviewing papers or reading papers, do you see a lot of examples of papers that have performed poor quality COMET?
Well, definitely, I would just say every time when I see the COMET papers, I will see whether they have positive control or negative control. It's very important to evaluate whether this COMET data is good or not. If they have a very good positive control, good positive control or negative control, they should using this control to stand up, to adjust the results, because every time the assay conditions are different. So the assay conditions control is very important. That's why in the nutrition study, we have 5 different control for the enzyme, like background control, treatment control, specific control, and traditional just for the strand break or something. So if you have a very good control group, you will have a very convincing results for the comet assay.
All right, thank you. Very good answer. I have one more question for you. And in your thesis, you briefly mentioned some other methods for assessing DNA damage. In ecotoxicology, we have a number of complementary methods for assessing DNA damage. And I would ask you to tell us if you know these methods that are commonly used for DNA damage assessment besides the comet assay.
Well, actually, they have many years ago, they also have one called alkaline elution assay. You are just, have a members just through, it's made by Gunnar Bonhoff, and just through that. But this one is time consuming. So we gave it up, go for the comet assay. Furthermore, they have a micronuclear test, but it's not for the DNA damage. It's for the micronuclear test. It's chromosome damage and break. Furthermore, it's gamma H2AX, but it's not for the single strand break. It's for double strand break, this one. But this gamma H2AX and the micronucleus test, it's a very good combinations with comet assay for the OECD, OECD risk assessment. Furthermore, they also have one called the Turner test, but that one is for the apoptosis. It cannot distinguish about apoptosis relative DNA damage or apoptosis relative DNA damage. Furthermore, they have one more. You can also use the PCR to do it, but the PCR is just like more DNA damage. That means it's inhibited the polymerization of the synthesis. So, but this one is also not only time consuming and it's not specific. And by the way, this one also needs the whole white of the DNA. So you need to isolate a very pure DNA from your samples. Could be a problem. I guess one should be the HPLC and HPLC, and I guess that one could be a very good one for measuring the DNA damage, especially for the 8-oxygen. But this one is already approved by the European standardized community on oxidative DNA damage. It can induce very high background of the oxidative Gauling, because they prove to the oxidations in the sample processing. I think probably, yeah, I think that's it.
I agree with you. I think you summarized very well. Thank you for your answers. This is very well responded. So I think I give the word back to the prorector.
Okay, thank you.
Thank you. The opposition will be continued by Professor Köhler, professor in anatomy at the Maastricht University.
Thank you, Prorector. Dear esteemed candidate, first my congratulations with your thesis. I'm from a completely different field, so I learned a lot, and there are probably a lot of things that I misunderstood or did not understand. In my imagination, I remember the comet assay from my youth, so I thought it was already very old, but I understand that it isn't that old actually. Definitely. But I was very curious. Imagine I have a very bad diet over a very long time, highly ultra-processed red meat or whatever, so a high risk of colon cancer because due to my bad habits. Or I am someone who has been exposed to bad drinking water acutely. With your comet assay and the DNA damage, could you see a difference can you distinguish between acute DNA damage and chronic DNA damage, or with a combination of assays?
So sorry, what type of the DNA damage? DNA damage?
Can you distinguish between acute damage? So let's say I've been exposed to toxic agents for a short period of time, or it has been bad lifetime choices for the last 20 years. So it's a chronic DNA damage.
Oh, yeah, I see. Well, sorry, dear highly esteemed opponent, thanks for your question. I had to say for this acute and chronic DNA damage, we cannot do that, but we can just we cannot using the DNA damage or DNA repair to figure it out separately. But this is one thing probably we can discuss is combine the DNA damage and the DNA repair. Probably this is one way to distinguish acute or chronic. It means for someone have a higher DNA repair, right? But these guys without the DNA damage data, you cannot distinguish whether this guy is really have a DNA repair ability from the genetic or something, or just in the high exposure. But when you combine these two parameters, you can distinguish whether this guy still exposure to the toxic agent and what's the real reactions of this human being. Well, if this guy is in the chronic exposure, right, could be these guys have a higher DNA damage, also have a higher DNA repair.
In the chronic situation.
Yeah, chronic situation.
And you would say in the acute situation, then the DNA repair response is not yet upregulated?
Yeah, could be.
Okay.
Yeah, just could be not that fast.
Okay. So would you then have to take several samples?
I think so. You know what? For us, the comet assay is no invasion test. We don't need a lot. Probably just a fingerprint, that's fine. Fingerprint, it just take 200 microliters and you can do the experiment. So I think for the human biometry, it's fine.
So you would always do it on PBMCs. You would not take, let's say, a bit more invasive. You say, I'm anyway busy with a coloscopy. I take, and it's been a exposure through my intestines. You would not take material from a coloscopy.
Well, it's you know what, we can use all the tissues or PBMC to do that. But I'm just talking about the human biomonitoring. But if, like you said, for the clinical trial, there is one group, they did a very good comet assay in the tissue. It's from the Pavlodica, from the chick. They just doing a lot of the comet assay experiment in the tissue.
Okay.
But for us, I just have some experiences in the muscles from the breast cancer patient. We can find this difference between the exposure and the no exposures group. But definitely, I think I need to think more about your question about whether this comet assay to distinguish the accurate DNA damage and the chronic DNA damage. It's a very good question, especially I'm thinking not only for the clinic, it's also a very good question for the human biomonitoring, because if this guy is chronic exposure to the toxic agent, could be this guy have a is already have a clonal expansion or inflammation, or is already in the process of the tumor genesis.
Okay. Oh, that's very good. And then you said something somewhere, and I don't know, I don't think it was your work, but you cited someone. You said if a patient has colon cancer and you have a biopsy, and then you find in the healthy tissue surrounding your biopsy, you find higher DNA repair with your COMET assay, that will be a good prognosis.
Yeah, this is.
Yeah. And what would you say if I find higher DNA or lower DNA repair in the tumor tissue itself? Would that also say something for the prognosis? Because you have both tissues.
This work is, like what I said, was did by the Pavlodica's group. They are just getting the samples from the tumors and also the adjacent tissue to do that. I'm thinking because so far it's unclear about the mutation in the DNA repair or DNA damage, the early stage to the cancer. But
Okay.
there is some study did by the Bevan and work in MIT. They are just trying to explain from the point mutation to the clonal expansion and to the tumorigenesis. I guess probably in this tumor tissue, they have some dysfunction of the DNA damage response in this one. But I don't know, and this so far is unclear how these tumor tissues just get bigger and make the adjacent tissue have a dysfunction. So it's a very good point to dig in the future, because if we really figure out this problem, we will understand the whole progress of the cancer. That will be a fascinating research. Probably deserve a Nobel Prize, right?
I think you have your work for the next 40 years. Thank
Thank
you for
you.
your answers. I give the word back to the pro-rector.
Thank you. The opposition will be continued by Professor van Zandvoort, Professor in Advanced Optical Microscopy at the Maastricht University. Professor van Zandvoort is also the secretary of this degree committee.
Dear Mr. Candidate, first of all, I would like to congratulate you, of course, you and your promotion team, and also your family and friends, with the thesis that was very well written, and also with this very important moment. But I specifically would like to congratulate you with chapter 2. That chapter, in my opinion, is exceptionally well written, and as a complete newbie in this field, I only had to read the chapter twice to become an expert. That means that I feel enough confidence to ask you some questions about that chapter.
Okay.
What I would like to focus on is your remark that you do visual scoring, and that actually you more or less think that is more convenient than image processing. You give some reasons for that, but as somebody who does a lot of microscopy imaging, I almost automatically go to automatic image processing. So could you shortly summarize why for you visual scoring, even at this moment, is still preferably?
Okay. Thank you, dearly highly esteemed opponent. Thanks for the question. Well, for this type of the questions, firstly, the visual scoring so far still have a 20% study utilize these methods. So that means still have many group without the resources could use the comet assay. That's why the one reasons we revise this guideline for them. Moreover, like you said, you are doing the automatic scoring microscopy or something. I had to say the visual scoring is a very important stage for the AI scoring, because you need to use, in the annotation stage, right? In your test data set, right? You need to use this guideline to do this annotation for different type of the comet for the deep learning and go for the CNN network models, right? So that's another reasons we still thinking about the visual scoring is a good thing. But the last thing is about when we compare the results between the visual scoring and semi-image scoring, right? Probably you have some mis-scoring because they believe about for the visual scoring, they are just arbitrary scoring. And for the semi-image scoring, they are not arbitrary scoring, but it's not. I think you just have a software in the computer. You just click by the investigator, so it's not a big difference. And we found the similar variabilities of the scoring in this two one. So definitely, I also collaborate with my colleague to make the automatic scoring. So I really think about this paper visual scoring helped me a lot to do the annotation work and doing the comet segregation stuff. I had to admit it is really helpful.
Yeah. So I do understand your argument that visual scoring is important anyhow, even if you do automatic image processing, because in the end you will need to do scoring.
Yeah.
That I understand. Your argument that image processing is also arbitrary with the current software and with the current possibilities of quantification and, for example, thresholding and segmentation and also then quantification, I think it is less arbitrary. I mean, everything is arbitrary in the end. I mean,
yeah, definitely.
but less arbitrary than visual scoring alone. So manual visual scoring. So let's put it like a contradiction between manual visual scoring and automated visual scoring. So could you speculate about a protocol to make the automatic image processing scoring more or less, basically less arbitrary and also faster? Because that's one of the arguments you use. It takes a lot of time.
Yeah. Well, I had to see, we already made the prototype of the automatic scoring in our part. So I had to see if you have these resources, definitely I will go for the automatic scoring because it's really save time. As you said, the threshold, the overlapping problems or something, it can solve the drawback of the semi-image scoring. But, for the visual scoring, I still believe about it's to our working methods because we should think about some experiment without the resources.
I understand that, but I think that you don't need very extensive resources for automated image processing. I do understand that you need visual scoring anyhow.
Yeah.
So that is obvious, or not visual scoring, but scoring. Don't you think also that, for example, going to a different way of imaging could help in getting better images that would be easier to score? And then I'm specifically thinking about confocal microscopy instead of wide field fluorescence microscopy. Also to look at the direction of the tail perpendicular to the plane of the imaging, so that you could, for example, look also at more complex angles and.
Oh yeah, you mean the ZX? Oh yeah, that could be interesting questions. There is one papers published by a group. I cannot remember the detail, but they are just using the tips in the slide and make the 3D comet. They can just 3D the axles. But what I can say is for our comet assays, the results we only need is personal DNA. The personal DNA calculation is total fluorescence of the tail divided of the total fluorescent tail. Even you have more versions, but the results are similar. And moreover, the things is about the two-photon microscopy, that is quite expensive, right?
No. I'm not talking about two-photon. I'm talking about confocal. Two-photon is a little bit too much, I think, for this.
For
also
us,
not needed.
Why we just go for the comet assay, like one of the esteemed opponents said, the comet assay is the old method. Why we insist on it? Because we are just trying to make it like automatic system for the fast screening, to help the government or health section to reduce the budget to screening the risk factor. That's the purpose we are doing, still insist on the comet assay. And moreover, you said probably just to try to get better images, or probably the images is not a big deal for the scoring. But the thing is about, there's one paper published by Roger Gastrock and Andrew Collin, Peter Moller, I guess it's last month, said the difference between for the comet assay, even a very small DNA damage could imply a biological implication. So we need to think about it.
And that means also that small changes could be very important.
Yeah, that's true.
Okay, I think my time is up. So I would like to give back to the project.
Thank you.
Thank you. The opposition will be continued by Dr. Skins, Associate Professor in Particle Toxicology at the Maastricht University.
Thank you, Prorector. Dear Mr. Candidate, I've been reading this thesis with very great interest, and I would like to congratulate you first, and also the team, and also congratulate your family and friends. And I work on particles a lot, so when I saw the thesis booklet itself, I was immediately thinking about seeing particles on the cover and interacting with DNA. And actually, that's a thing what I would like to discuss with you about a little bit about beyond your current data. So I've noted in your publication list that you also worked a little bit on nanomaterials in vitro. And you know there is a discussion on its relevance in terms of DNA damage investigation. But I wonder what your opinion is about the applicability of your DNA repair assays when you test nanomaterials, engineered nanomaterials or advanced materials. Are we already okay there, or is there still a lot to do in this regard?
Okay, thank you, dear highly esteemed opponent. Thanks for your question. Actually, this is one project I'm talking with my supervisors, I guess 1 year ago. We are probably just applied a project for the nanomaterial to whether the influence on the DNA repair, because most of the research, they are focused on the DNA damage after treated with the nanomaterial. Barely research go for the DNA repair. We're thinking about it's a very interesting project we can go for. What I can see is for the nanomaterial, it's very complicated to evaluate because they have some factors you need to consider about it. The size, the property, and the shape, the differences. There is one paper we collaborate with the Norwegian Air Quality Institute. It's just like how to evaluate and how to report the nanomaterial for DNA damage. So I will also follow the evaluation strategy or strategy to measuring the DNA repair after treated with the nanomaterial. If I have the resources, I will go for that.
Okay. So you know that many of these nanomaterials, they are going into cells. And I was just wondering, you're working with lysates in your in vitro repair assay. Don't you get a problem there that all these proteins are absorbed also on all these internalized nanomaterials? Or do you have any idea how to deal with that? Or
well, it's I'm just thinking there is one, like you said, there is one papers called microplastic and the nanoplastic. They can just absorb it in the human body, even in the testicles. So yeah, there is one more project I will go for in the future. Is just to evaluate human infertility after nanoplastic or microplastic digestion in the testicle samples in male. But what I can say is probably there is there is one way, or using another nanomaterial to go for the cells to react with the exposure of nanomaterial, the bad nanomaterial, to take them out from the body. I'm not sure about that. I just speculate it. I'm not very good in this in vivo stuff.
So a little bit more into the mechanism. Do you think that actually when particles go into the cell that they could have an impact on the DNA repair? Could they mess up with repair, or could it be realistic not only for your testing system but also in the real life situation? You mentioned a paper, for instance, by Peter Muller and others, where they looked at air pollution and looked at DNA damage. And there's a lot of nanoparticles in the outside air also, which we inhale. Do you think that could be a problem in terms of the interpretation? Did they actually measure DNA damage, or did they measure repair?
I'm thinking because firstly, I should be clarified the DNA damage and the DNA repair, they are not independent pathway. They belongs to the DNA damage response. So you should think about whether this nanomaterial, the concentration, is toxic, genotoxic or not. If it is genotoxic, that means the DNA damage response. Oh, sorry. So
Congying Zheng, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return.
Okay, thank you. And can I just continue for a little bit? I just want to finish the question.
No, it's finished.
Yes, we are all back, Pro-Rector. Go ahead.
Thank you. Congying Zheng, the degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor van Schoten is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Thank you.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Congying Zheng, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate, signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, shown by the beadle. You can see it there.
Thank you.
Congratulations, Dr. Zheng. You have delivered an outstanding achievement today, an impressive dissertation, and a strong defense today. Big compliments. And now Dr. Gottschalk will deliver some words to you and pronounce what we call here the laudatio. Roger, the floor is yours.
Thank you. So, Dr. Zheng, today we are not only celebrating a remarkable academic achievement, but we are also a journey across many borders, a journey that tells your story as a true world citizen. You are originally from China, but you took your first steps into international academia in Hungary, where you did your master. And then you pursued your PhD in the Netherlands, but you did your research in the breathtaking landscapes of Norway. And now you set sail for a new horizon into the United States at the MIT in Boston. I think few people have so many homes. In a time where the world often seems to be drawing lines between countries, between ideologies, and even between people, it is incredibly powerful to witness a story like this. This is your story. A story that reminds us that research should have no borders and that curiosity is stronger than division. It shows us that collaboration is richer when diverse minds come together. And I have to admit that you had to deal with a lot of diverse minds. All your supervisors have strong opinions, and they had their own ideas about what direction your work should go to. Andrew, Gunnar, Friedrich-Jan, Sergey, me, and yourself. Often we could have long discussions on relatively simple things, like how to define a single word in a paper. I still remember the discussions on how to define test cells, substrate cells, or substrate nucleoids, or another discussion in which we had to decide whether we should use DNA repair capacity or DNA repair activity, or is it maybe incision capacity or incision capacity? We still don't know what is best. Long discussions, but you managed to guide your project through, and today you successfully defended your thesis. I especially appreciate that you are full of ideas, and you will continue to work on part of those ideas in the US. But you have to keep in mind that you only have 24 hours in a day and 7 days in a week. But somehow you seem to have more time than others, because you may have a few chapters in your thesis, but you authored and co-authored a total of 14 papers already, and more is in preparation. So your scientific journey is far from over. More is yet to come. May this scientific journey ahead continue to build bridges in a world that seems to need them. May your work remind us all that the best idea, the strongest solution, the brightest future comes when we reach out across languages, across cultures, and across continents. So congratulations, Dr. Zheng. You carry many flags, but more importantly, you carry a future in science. Congratulations.
Okay, thank you.
Dear Dr. Zheng, also on behalf of the Board of Deans of Maastricht University, it's a pleasure for me to congratulate you with the degree you just have acquired. It's already expressed by your co-supervisor. We are expecting a lot of your future research, but the enthusiasm during the defense is, I believe, a very good basis for future positive outcomes and productivity and creativity. So we wish you all the best in your future career. And hereby I close this academic ceremony.
Okay, thank you, dear